The concept of de-extinction. De-extinction refers to the process of bringing back extinct species using modern science. The idea relies on advanced genetic technologies to recreate or reintroduce species that have vanished. While it sounds like science fiction, de-extinction is a topic of serious scientific inquiry and debate. DNA retrieval and analysis. The first step in de-extinction is obtaining and analyzing DNA from extinct species. Advances in genetic sequencing have enabled scientists to extract DNA from fossils or preserved specimens. This DNA provides a blueprint for reconstructing the genetic makeup of the extinct species, although it is often degraded and incomplete. Cloning extinct species. Cloning involves creating a genetically identical organism from a DNA sample. For extinct species, scientists would use DNA from well-preserved remains and insert it into the egg cells of closely related species. For example, the woolly mammoth might be cloned using the DNA from mammoth remains and Asian elephant egg cells. However, successful cloning of extinct species remains a significant challenge. Genetic engineering and editing. Genetic engineering techniques, such as CRISPR, offer another approach to de-extinction. By editing the genome of a closely related species, scientists can introduce traits from the extinct species. For instance, researchers are working on modifying the genes of modern elephants to recreate traits of the woolly mammoth. This method involves less direct cloning but requires precise genetic modifications. Ethical considerations. De-extinction raises numerous ethical questions. Concerns include the welfare of resurrected animals, potential ecological impacts, and the cost versus benefit of such projects. Ethical debates also consider whether humans should intervene in natural processes and the implications of reintroducing species into modern ecosystems. Ecological impacts and habitat suitability. Reintroducing extinct species could have unpredictable ecological impacts. The environment has changed significantly since these species went extinct, and their reintroduction might disrupt existing ecosystems or fail to meet the species' needs. Scientists must consider habitat suitability and potential interactions with current flora and fauna. Success stories and challenges. While some de-extinction projects have shown promise, success stories are limited. The resurrection of the Pyrenean ibex, which briefly existed before its second extinction, highlights both the potential and the challenges of de-extinction. The process requires overcoming numerous scientific, logistical, and ethical hurdles. Conservation and de-extinction. De-extinction could play a role in conservation efforts, helping to restore lost biodiversity and ecosystems. However, it is not a substitute for protecting existing species and habitats. Conservationists argue that resources might be better spent on preserving current species and their environments. Future prospects. The future of de-extinction depends on advances in genetic technology, improved understanding of extinct species biology, and careful consideration of ethical and ecological factors. Ongoing research may eventually make de-extinction more feasible, but it will require interdisciplinary collaboration and rigorous assessment. Conclusion. Resurrecting extinct species with DNA is a fascinating and complex field of science with both potential and challenges. While progress in genetic technology offers hope, the practical, ethical, and ecological aspects must be carefully considered. De-extinction presents opportunities for scientific advancement but must be approached with caution and responsibility.